Hey guys, welcome to the VFitter podcast. This is a podcast that's dedicated to learning about AI integrations, industry-specific applications, and looking at super cool AI trends in the industry today. Today, we're going to be exploring the intersection between neuroscience and AI, as well as industry-specific applications for AI in the context of our platform and biotechnology. In today's video, we're going to be exploring Mark Zuckerberg and Dr. Priscilla Chan's recent interview with Andrew Huberman as the intro video and the first video that we're going to react. After this, we are going to be introducing our guest speaker for today, Dr. Shaina Cahill, who is a PhD holder and a neuroscientist based out of Lethbridge, Alberta. We're going to be discussing the commonplace applications for how AI can be integrated into scientific workflows, research in the context of drug discovery, neuroscience, and biotechnology as an industry whole, as well as explore those applications specific to the VFitter platform and how we are inventing new research methods for biotech with AI on our platform today. And last but not least, our podcast will be moderated and hosted by Elijah Royaei, who is the founder of VFitter, our all-in-one AI cloud custom workplace.
Thank you so much, Nick.
Okay, let's get started.
Everyday life. I'm Andrew Huberman, and I'm a professor of neurobiology and ophthalmology at Stanford School of Medicine. My guests today are Mark Zuckerberg and Dr. Priscilla Chan. Mark Zuckerberg, as everybody knows, founded the company Facebook. He is now the CEO of Meta, which includes Facebook, Instagram, WhatsApp, and other technology platforms. Dr. Priscilla Chan graduated from Harvard, and funding not available elsewhere, as well as a novel framework for discovery of the basic functioning of cells, cataloging all the different human cell types, as well as providing AI or artificial intelligence platforms to mine all of that data to discover new pathways and cures for all human diseases. Priscilla Chan. Priscilla, Mark, so great to meet you, and thank you for having me here in your home.
Oh, thanks for having us on the podcast.
Yeah.
I'd like to talk about the CZI, the Chan-Zuckerberg Initiative. I learned about this a few years ago when my lab was and still is now at Stanford as a very exciting philanthropic effort that has a truly big mission. I can't imagine a bigger mission. So maybe you could tell us what that big mission is, and then we can get into some of the mechanics of how that big mission can become a reality.
So, like you're mentioning, in 2015, we launched the Chan Zuckerberg Initiative. And what we were hoping to do at CZI was think about how do we build a better future for everyone, and looking for ways where we can contribute the resources that we have to bring philanthropically and the experiences that Mark and I have had, for me as a physician and educator, for Mark as an engineer, and then our ability to bring teams together to build builders. Mark has been a builder throughout his career, and what could we do if we actually put together a team to build tools, do great science? And so within our science portfolio, we've really been focused on what some people think is either an incredibly audacious goal or a inevitable goal, but I think about it as something that will happen if we sort of continue focusing on it, which is to be able to cure, prevent, or manage all disease by the end of the century.
All disease.
All disease. So that's important, right? And a lot of times people ask, like, which disease? And the whole point is that there is not one disease. And it's really about taking a step back to where I always found the most hope as a physician, which is new discoveries and new opportunities and new ways of understanding how to keep people well come from basic science. So our strategy at CZI is really to build tools, fund science, change the way basic scientists can see the world and how they can move quickly in their discoveries. And so that's what we launched in 2015. We do work in three ways. We fund great scientists. We build tools, right now software tools, to help move science along and make it easier for scientists to do their work. And we do science. You mentioned Stanford being an important pillar for our science work. We've built what we call biohubs, institutes where teams can take on grand challenges to do work that wouldn't be possible in a single lab or within a single discipline. And our first biohub was launched in San Francisco, a collaboration between Stanford, UC Berkeley, and UCSF.
Amazing.
Cool.
Curing all diseases.
Yeah, this is just like, do you want to start?
Yeah, so I think that their take on how kind of to approach science and how it kind of highlights how science is changing, both from a not just governmental funding. I think there's more of a reliance now outside of government funding, but as well as this idea that there's an importance to basic science, which I think for a long time was kind of overlooked. It's a little bit more of a nitty gritty, but also supporting basic science allows for scientific growth. And I think that's a really cool aspect to see from someone who wants to cure a disease, to understand that really that starts at really understanding like what the disease is, and coming at it through these kind of hubs are bringing a lot of people together to answer the question. We're seeing a shift in medicine in that way as well, with more functional medicine, which is looking at, a disease as a whole or as a person as a whole. And how does that help us better answer questions? And so we see in a lot of diseases, there's kind of a shift in understanding, or we're starting to look at diseases from a wider scope. And I think this kind of initiative kind of helps support that, which is, bringing people together that are all studying something similar, actually help us put the puzzle together quicker than each of us looking at each piece individually. So I think it's kind of a really exciting initiative. While it's a big project to kind of cure all diseases, I think we're starting to learn that, diseases overlap a lot. And so getting a better understanding of the whole puzzle seems to be maybe the better route to go through it. Cool. But the other thing, now related, the other thing about what they're doing is by bringing two people together that come from different sources of information, you're seeing they're able to better connect kind of technology with science. And, as technology grows, I think utilizing that allows us to get answers quicker, and kind of doing that in the right way. So working with scientists or finding out how technology could help versus creating a product and saying this product will help, working with scientists to figure out where are the areas where you feel like this could be faster? How do we best utilize, these are the skills we have as an engineer, as someone that, understands computers. This is what we could do. How does that work for you? And working in conjunction, I think allows for better growth and better utilization, because you're custom making something to help them do what they're, whatever their end goal is, versus what traditionally is the case is where you buy a software and you try to make it work for you. Having someone help you create a software that works perfectly for you and making that the norm, I think will help kind of both integrate those fields together quicker, but also help actually accelerate things by having products that work for you.
Yeah, I totally agree, because Mark Zuckerberg, they're actually like a power couple. But Mark Zuckerberg have been working on entertainment stuff, right? So it's like technology in channel of like entertainment. But now that it's mixed with science, it's now helping humanity way more, because like social media and all this stuff is like addictive, right? So a lot of people hating on him just to have this addictive platform and like dragging everyone into it. But now he's trying to like basically do it in like a way that it benefits humanity.
Yeah. And I think we need to understand that maybe social media has grown more than we had expected it to, but there are a lot of positives to it. There are a lot of groups and people that can't connect as easily, and allowing people to live in multiple places and connect with each other. Like social media has a lot of positives. There was a lot of science that went into social media. There's a lot of psychology. There's a lot of neuroscience and stuff that goes into why social media works the way it works and why people like it. But like everything, when you get kind of mass appeal, there's negatives and positives with it. So, but I do like seeing like they have a lot of money and they have a lot of clout on their own. So putting that towards focusing the large population, using who they are on, like let's use technology for good, I think is also, really admirable.
Exactly. Okay, let's keep watching.
That there will either be a ton of knowledge gleaned from this effort, which I'm certain there will be, and there already has been. We can talk about some of those early successes in a moment. But it also sort of implies that if we can understand some basic operations of diseases and cells that transcend autism, Huntington's, Parkinson's, cancer, and any other disease, that perhaps there are some core principles that would make the big mission a real reality, so to speak. What I'm basically saying is, how are you attacking this? My belief is that the cell sits at the center of all discussion about disease, given that our body is made up of cells and different types of cells. So maybe you could just illuminate for us a little bit of what the cell is in your mind as it relates to disease, and how one goes about understanding disease in the context of cells, because ultimately that's what we're made up of.
Yeah, well, let's get to the cell thing in a moment, but just even taking a step back from that, we don't think that at CZI that we're going to cure, prevent, or manage all diseases. The goal is to basically give the scientific community and scientists around the world the tools to accelerate the pace of science. And we spent a lot of time when we were getting started with this looking at the history of science and trying to understand the trends and how they've played out over time. And if you look over this very long-term arc, most large-scale discoveries are preceded by the invention of a new tool or a new way to see something. And it's not just in biology, right? It's like having a telescope, came before a lot of discoveries in astronomy and astrophysics. But similarly, the microscope and just different ways to observe things, or different platforms, like the ability to do vaccines preceded the ability to kind of cure a lot of different things. So this is sort of the engineering part that you were talking about building tools. We view our goal is to try to bring together some scientific and engineering knowledge to build tools that empower the whole field. And that's sort of the big arc, and a lot of the things that we're focused on, including the work in single cell and cell understanding, which you can jump in and get into that if you want. But yeah, I think we generally agree with the premise that if you want to understand this stuff from first principles, people study organs a lot, right? They study kind of how things present across the body, but there's not a very widespread understanding of how kind of each cell operates. And this is sort of a big part of some of the initial work that we tried to do on the Human Cell Atlas and understanding what are the different cells, and there's a bunch more work that we want to do to carry that forward. But overall, I think when we think about the next 10 years here of this long arc to try to empower the community to be able to cure, prevent, or manage all diseases, we think that the next 10 years should really be primarily about being able to measure and observe more things in human biology. There are a lot of limits to that. It's like you want to look at something through a microscope. You can't usually see living tissues because it's hard to see through skin or things like that. So there are a lot of different techniques that will help us observe different things. And this is sort of where the engineering background comes in a bit, because when I think about this is from the perspective of like how you'd write code or something, the idea of like trying to debug or fix a code base but not be able to like step through the code line by line, it's like never not going to happen, right? And at the beginning of any big project that we do at Meta, we like to spend a bunch of the time up front just trying to instrument things and understand what are we going to look at and how are we going to measure things before so we know we're making progress and know what to optimize. And this is such a long-term journey that we think that it actually makes sense to take the next 10 years to build those kind of tools for biology and understanding just how the human body works in action, and a big part of that is cells. I don't know. Do you want to jump and talk about some of the efforts?
Could I just interrupt briefly and just ask about the different interventions, so to speak, that CZI is in a unique position to bring to the quest to cure all diseases. So I can think of, I know as a scientist that money is necessary but not sufficient, right? Like when you have money, you can hire more people, you can try different things. So that's critical. But a lot of philanthropy includes money. The other component is, you want to be able to see things, as you pointed out. So you want to know the normal disease process. Like what is a healthy cell? What's a diseased cell? Are cells constantly being bombarded with challenges and then repairing those? And then what we call cancer is just kind of the runaway train of those challenges not being met by the cell itself or something like that. So better imaging tools. And then it sounds like there's not just a hardware component, but a software component. This is where AI comes in. So maybe we can, at some point, we can break this up into three different avenues. One is understanding disease processes and healthy processes. We'll lump those together. Then there's hardware, so microscopes, lenses, digital deconvolution, ways of seeing things in bolder relief and more precision. And then there's how to manage all the data. And then I love the idea that maybe AI could do what human brains can't do alone, like manage understanding of the data. Because it's one thing to organize data. It's another to say, this, as you point out in the analogy with code, that this particular gene and that particular gene are potentially interesting, whereas a human being would never make that potential connection.
So, the tools that CZI can bring to the table, we fund science, like you're talking about. And we try to, there's lots of ways to fund science. And just to be clear, what we fund is a tiny fraction of what the NIH funds, for instance. So
although you guys have been generous enough that it definitely holds weight to NIH's contribution.
Yeah, but I think every funder has its own role in the ecosystem. And for us, it's really how do we incentivize new points of view? How do we incentivize collaboration? How do we incentivize open science? And so a lot of our grants include inviting people to look at different fields. Our first neuroscience RFA was aimed towards incentivizing people from different backgrounds, immunologists, microbiologists, to come and look at how our nervous system works and how to keep it healthy. Or we asked that our grantees participate in the preprint movement to accelerate the rate of sharing knowledge and actually others being able to build upon science. So that's the funding that we do. In terms of building, we build software and hardware, like you mentioned. We put together teams that can build tools that are more durable and scalable than someone in a single lab might be incentivized to do. There's a ton of great ideas, and nowadays most scientists can tinker and build something useful for their lab. But it's really hard for them to be able to share that tool sometimes beyond their own laptop or forget, the next lab over or across the globe. So we partner with scientists to see what is useful, what kinds of tools, in imaging, Napari, it's a useful image annotation tool that is born from an open source community. And how can we contribute to that? Or Cell by Gene, which works on single cell data sets, and how can we build a useful tool so that scientists can share data sets, analyze their own, and contribute to a larger corpus of information. So we have software teams that are collaborating with scientists to make sure that we're building easy to use, durable, translatable tools across the scientific community in the areas that we work in. We also have institutes. This is where the imaging work comes in, where, we are proud owners of an electron microscope right now. It's going to be installed at our imaging institute, and that will really contribute to a way where we can see work differently. But the more hardware does need to be developed. We're partnering with a fantastic scientists in the biohub network to build a mini faceplate to align the electrons through the electron microscope to be able to increase the resolution so we can see in sharper detail. So there's a lot of innovative work within the network that's happening. And these institutes have grand challenges that they're working on. Back to your question about cells. Cells are just the smallest unit that are alive. And your body, all of our bodies have.
Yeah. So before she gets into the cell stuff, I think relevant to VFitter, what you guys are doing, talking about these big data sets or open access information or easily accessible information. It's really great that there's so much out there. From my perspective, as a neuroscientist, the Allen Brain Institute has a ton of information you can access. There are lots of platforms, there's lots of databases that have plethoras of information that's gathered. either has been created or has been created amongst what's been done in the research. But the downside with some of these cases is that, like, what do you do with that? How do you, like, there's so much data. There was a big movement for a very long time, still exists, in big data. We just want more data. We need more data. That's completely correct. The downside is when you have a lot of data, how do we process it? How do we best look at it? How do you parse it up? How do you put it together? How do you take two databases and make them interact so that you get the right questions? And I think that's where these softwares come in. This is where adaptive software like AI comes in, because it can take these big data sets and you can ask it questions and can integrate them together. It can look across, 10 databases at the same time and filter based on the viewpoint you're looking at. And in some cases, as one person or one lab or one institute, it is really hard to take all that big data and really use it, I think, in its best way, package it properly so that you can filter it and understand it and come ahead because of that data. So I think the idea of using technology in a software sense to better utilize data that's been created from, these more, new microscopes, more large scale, clinical trials, taking all of that information together from the cell to the person, but then being able to put those together and ask questions using AI, which can then filter through all of this stuff and then kind of come up with new conclusions or further questions, I think is really exciting, because big data is huge and it's so important, but it is somewhat daunting to figure out, like, how do we go about using it the best way possible?
Well, we ask AI.
Yeah.
We ask, how do we use it in the best way?
And then I think the other point she made was that working with scientists to make softwares that are easy to use and aren't intimidating. It's hard sometimes to picture someone who can use an electron microscope, which is like one of the most complicated things, or do these really high-tech scientific, experiments, but at the same time would have trouble creating a software that works for them. So, there's different versions of SMART. There's also different versions of kind of leaning into your skill set. So by taking someone who has a really high skill set in AI and someone who has a really, high skill set in cell biology and getting them to work together allows you to make the best tool possible, because it's utilizing I'm able to make AI do this, but then what do you need it to do? And then the other person's able to really have a full scope of understanding of what is the question, what's the most important information, what are the rules that it has to follow to get you the right information. By putting those two people together, you make something that, works seamlessly with what's already happening in the lab. And that's kind of part of what it is when you're working in biotech, biopharma science, is that there's already a process happening to get that piece of data that you're going to look at. You want the tool you use that analyzes the data to have an understanding or come at it from the right way, with the understanding of what's happening kind of at the bench, or, what's happening when you're kind of analyzing a drug, that kind of stuff. So working together in that way, I think, creates the best tool, and then creates a tool that's easy to use. So then it's actually utilized to its fullest potential. And I think that's important, is making it easy to use so that people do use it.
Exactly. As you said, a lot of these databases are publicly available. Most of them are open source, so it's like free. But like a lot of people don't know how to use it. And there's also like these large language models, they target the information vertically. Like I call them, like, it's like you give 10 books, but then you ask for a table that categorizes those books all in one table, right? So that's like really targeting the knowledge as you want it. And a lot of these like databases are cool, they're out there, but just think about a data collection tool that is being invented right now. They're going to talk about it, and it's going to go inside the cell, and it's going to gather information based on the cell, like whatever is happening inside the cell, and the relationship with the whole organ that is happening. And if AI processes that, that's like you have lifetime, like you have live information, and you could process them. You could predict diseases. You could predict cancer, potentially even cure it. But yeah, it's super cool. So I'm starting.
Many cells. There's, some estimate of like 37 trillion cells, different cells in your body. And what are they all doing? And what do they look like when they're healthy? When you're healthy, what do they look like when you're sick? And where we're at right now with, our understanding of cells and what happens when you get sick is basically we've gotten pretty good at, from the Human Genome Project, looking at how different mutations in your genetic code lead for you to be more susceptible to get sick or directly cause you to get sick. So we go from a mutation in your DNA to, wow, you now have Huntington's disease, for instance. And there's a lot that happens in the middle. And that's one of the questions that we're going after at CZI is what actually happens. So an analogy that I like to use to share with my friends is right now, say we have a recipe for a cake. We know there's a typo in the recipe. And then the cake is awful. That's all we know. We don't know how the chef interprets the typo. We don't know what happens in the oven. And we don't actually know sort of how it's exactly connected to how the cake didn't turn out how you had expected. A lot of that is unknown, but we can actually systematically try to break this down. And one segment of that journey that we're looking at is how that mutation gets translated and acted upon in your cells. And all of your cells have what's called mRNA. mRNA are the actual instructions that are taken from the DNA. And what our work in single cell is looking at how every cell in your body is actually interpreting your DNA slightly differently. And what happens when healthy cells are interpreting the DNA instructions and when sick cells are interpreting those directions. And that is a ton of data. I just told you there's 37 trillion cells. There's different large sets of mRNA in each cell. But the work that we've been funding is looking at how, first of all, gathering that information. We've been incredibly lucky to be part of a very fast-moving field where we've gone from in 2017 funding some methods work to now having really not complete, but nearly complete atlases of how the human body works, how flies work, how mice work at the single cell level, and being able to then try to piece together, like, how does that all come together and when you're healthy and when you're sick? And the neat thing about the sort of inflection point where we're at in AI is that I can't look at this data and make sense of it. There's just too much of it. And biology is complex. Human bodies are complex. We need this much information. But the use of large language models can help us actually look at that data and gain insights. Look at what trends are consistent with health and what trends are unsuspected. And eventually, our hope through the use of these data sets that we've helped curate and the application of large language models is to be able to formulate a virtual cell, a cell that's completely built off of the data sets of what we know about the human body, but allows us to manipulate and learn faster and try new things to help move science and then medicine along.
Do you think we've cataloged the total number of different cell types? Like, every week I look at great journals like Cell, Nature, and Science. And for instance, I saw recently that using single cell sequencing, they've categorized maybe 18 plus different types of fat cells. We always think of like a fat cell versus a muscle cell. Mm-hmm. So now you've got 18 types. Each one is going to express many, many different genes and mRNAs. And perhaps the one of them is responsible for what we see in advanced type 2 diabetes or in other forms of obesity, or where people can't lay down fat cells, which turns out to be just as detrimental in those extreme cases. So now you've got all these lists of genes, but I always thought of single cell sequencing as necessary but not sufficient. Right? You need the information, but it doesn't resolve the problem. And I think of it more of hypothesis generating experiment. Like so you have all these genes and you could say, wow, this gene is particularly elevated in the diabetics cell type of, let's say, one of these fat cells or muscle cells for that matter, whereas it's not in non-diabetics. So then of the millions of different cells, maybe only 5 of them differ dramatically. Mm-hmm. So then you generate a hypothesis, it's the ones that differ dramatically that are important, but maybe one of those genes, when it's only, 50% changed, has a huge effect because of some network biology effect. And so I guess what I'm trying to get to here is, how does one meet that challenge? And can AI help resolve that challenge by essentially placing those lists of genes into a 10,000 hypotheses? Because I'll tell you that the graduate students and postdocs in my lab get a chance to test one hypothesis at a time. And that's really the challenge, let alone one lab. Yeah. And so for those that are listening to this, and hopefully it's not getting outside the scope of kind of like standard understanding or the understanding we've generated here, but what I'm basically saying is you have to pick at some point. More data always sounds great, but then how do you decide what to test?
So no, we don't know all the cell types. I think one thing that was really exciting when we first launched this work was, cystic fibrosis. Like cystic fibrosis is caused by a mutation in CFTR. That's pretty well known. It affects a certain channel that makes it hard for mucus to be cleared. That's the basics of cystic fibrosis. When I went to medical school, it was taught as fact.
So their lungs fill up with fluid. These are people carrying around sacks of fluid filling up. I've known people, I've worked with people like that, and they have to literally dump the fluid out.
Exactly.
They can't run or do intense exercise. Life is shorter.
Life is shorter.
Yeah.
And when we applied single cell methodologies to the lungs, they discovered an entirely new cell type that actually is affected by a mutation in the CF mutation and cystic fibrosis mutation that actually changes the paradigm of how we think about cystic fibrosis.
Amazing.
Just unknown. So I don't think we know all the cell types. I think we'll continue to discover them, and we'll continue to discover new relationships between cell and disease. Which leads me to the second example I want to bring up is this large data set that the entire scientific community has built around single cell is starting to allow us to say, this mutation, where is it expressed? What types of cell types it's expressed in? And we actually have built a tool at CZI called Cell by Gene, where you can put in the mutation that you're interested in, and it gives you a heat map of cross cell types of which cell types are expressing the gene that you're interested in. And so then you can start looking at, okay, if I look at gene X and I know it's related to heart disease, but if you look at the heat map, it's also spiking in the pancreas. That allows you to generate a hypothesis. Why? And what happens when this gene is mutated in the function of your pancreas? Really exciting way to look and ask questions differently. And you can also imagine a world where if you're trying to develop a therapy, a drug, and the goal is to treat the function of the heart, but you know that it's also really active in the pancreas again. So is there going to be an unexpected side effect that you should think about as you're bringing this drug to clinical trials? So it's an incredibly exciting tool, and one that's only going to get better as we get more and more sophisticated ways to analyze the data.
I must say I love that, because if I look at the advances in neuroscience over the last 15 years, most of them didn't necessarily come from looking at the nervous system. Came from the understanding that the immune system impacts the brain. Everyone prior to that talked about the brain as an immune privileged organ. What you just said also bridges the divide between single cells, organs, and systems, right? Because ultimately cells make up organs, organs make up systems, and they're all talking to one another. And everyone nowadays is familiar with like gut-brain axis or the microbiome being so important, but rarely is the discussion between organs discussed, so to speak. So I think it's wonderful. So that tool was generated by CZI, or CZI funded that tool. So how does this?
We built that.
We built it.
You built it. So is it built by Meta? Does Meta have a hand?
CZI has its own engineers.
Got it.
Yeah. They're completely different organizations.
And so a graduate student or postdoc who's interested in a particular mutation could put this mutation into this database. That graduate student or postdoc might be in a laboratory known for working on heart, but suddenly find that they're collaborating with other scientists that work on the pancreas.
Yep.
Which also is wonderful because it bridges the divide between these fields. Fields are so siloed in science, not just different buildings, but people rarely talk unless things like this are happening.
I mean, the graduate student is someone that we want to empower because one, they're the future of science, as you know. And within Cell by Gene, if you put in the gene you're interested in and it shows you the heat map, we also will pull up like the most relevant papers to that gene. And so like read these things.
Fantastic. As we all know, quality
I know one question. Now the graduate student has questions. So do you think all these like cell types that we haven't really discovered them yet, do you think it's different from body to body? Like, for example, for my cells, like I could have 30 different types and then you could have the 30 other different types and like you couldn't find those in my body, or are they the same? And the second question is like, are we going to be able to help finding more cell types with AI? Do you think that's possible?
Yeah. So I wouldn't say there would be distinct, like your body isn't distinctly made up different than my body. The cells are pretty consistent. Every organ is made up of a bunch of different types of cells. Your blood has different types of cells. So the building blocks are generally the same. That's a general overview. There may be some differences, how different cells are expressed, how many of each kind could change. But when they're talking, about things like mRNA, what we're saying is that within each cell, they have like a library within each cell. How many pages in each book could be different. How often each book is read is different between each person. So things like your genetics play a really big role, but also what food you eat plays a big role. That can then impact how cells work. The food you eat is the building blocks of everything else within your body. Also, your environment plays a big role. Fun example, I do a lot of work right now with the gut microbiome, gut-brain connection. And so we see when there's been studies that done that basically when people live in one environment and eat one type of food, their body functions in one way. Their gut microbiome looks a certain way, the types of vitamins they produce, all that kind of stuff, it looks one way. When they move to westernized culture, where a Western diet is more predominant, which is higher in fat, lower in fiber, higher in sugars, processed foods, things like that, their gut microbiome and how their gut functions and what they're more likely to, like their likelihood of occurring certain diseases actually shifts within 6 months of moving. So your environment, what you're putting into your body plays a huge role in how your body functions. So everybody is different based on what they're doing to their body, what environments they put them in, what they're feeding their bodies, how much exercise they're doing, how much sleep, all of those things play a big role. And we also know it kind of builds on itself, right? So if you are someone who exercises a lot, you tend to eat healthy. Well, those two things are actually related as well. If you eat really poorly, it can impact how likely you are to get a good night's sleep, what your sleep then functions on, how likely you are to exercise, how social you're going to be. That can then impact your mental health. So all of these things are included, and all of that starts with what you're feeding the cells and what the cells are able to do. And that can shift over time. Also, the flexibility of our body to repair itself changes with age. So as we age, different things become more important. And when they make the point of like, while single cell stuff is really important and it gives us a lot of information, the story is quite large. So each piece of this little piece of information helps us build to understand, a cell that helps us understand how an organ functions. An organ's functioning allows us to better understand how two organs communicate, which then allows us to understand how a system functions. And then once we understand that system, then we can start understanding what impacts from outside the system and what happens when the system malfunctions. So it's such a these concepts are quite large, and we're learning more and more. And one of the things that I used to talk about when I was still a graduate student, and I would talk to other graduate students, was it almost feels like once you almost have an answer, you learn something that shifts your perspective on the whole story again, and you kind of had to start back at the beginning. And if you think, they were talking a lot about how scientific discoveries either new technologies or new abilities to look at things can change how we understand science. And he made a good point about how for a long time we thought the brain, as a brain person, for a very long time, neuroscientists kind of were like, the brain does everything and nothing else matters. And kind of all science areas had a tendency to be like that. They're very independent, don't tend to work well together. And that's shifting, which is really important. But you have to think about things like we didn't know the immune system could impact the brain. This is a specific cell type that really kind of is the driver of immune function within the brain. But we're learning now that, the gut can cause inflammation, but that gut inflammation then can impact the amount of inflammation in the brain. And so it's a bigger system. But now that we understand that, when we start thinking about autoimmune disorders in the brain or disorders of the brain that are triggered or heighten inflammation, is that only the brain or something else happening in the body that's impacting the inflammation in the brain? So are diseases just brain diseases? Probably not. They're probably whole system problems. And so we're kind of evaluating how we look at things. You have to think about, there was a period of time we didn't know what DNA was. And after that, we didn't know what mRNA was. And and then we learned what, epigenetics was. And so I always think about we learned what DNA was, which is kind of what we thought decided everything. And all of a sudden we learned what epigenetics was, which is basically how genes are expressed. So all of a sudden, when we talk about environmental factors, epigenetics, we know, can be influenced from your parents' parents, and as far back as your great-great-grandparents, can impact how your genes are displayed. So then it becomes a much more complicated thought, because it's not just the DNA or the genes, it's also how are they expressed, when are they expressed, what impacts that. And then as our environment changes, the environment plays a role. So it's a very big story, but they make a good point of we have to understand the building blocks of all of that. And then, now we're learning those building blocks. Well, okay, so this gene's malfunctioning in this disease, but also now they're able to tell us, well, it's not just in the heart, it's in this other organ as well. So are those two independent things? Are they impacting each other? There is a lot of chicken and egg in science, which comes first, we don't know.
Yeah.
But better understanding that, you can't just function. It's not just one thing. It could be 5 things. How do we look at the story from all 5 of those things being involved? Is that actually going to get us further along? And so by being able to kind of see big data in the sense of is there more to the story than the viewpoint I have? Big data allows us to see that, because we can see from outside of, our kind of tunnel vision. As a brain person, you think about the brain, but it's not just the brain. There's a bunch of stuff happening. And so big data and the ability to kind of integrate a bunch of data sets from different areas together allows us to have a bigger viewpoint. And I think better answer questions, but more importantly, ask better questions that leads to the next, scientific study. Because it's not just all what the answer is. It's so what's the next question that gets us to the next piece of data that helps us actually answer that question? Because we know a lot already, but we're not very far ahead in a lot of things. I spent a lot of time looking at Alzheimer's disease and in that realm. And one of the things that we would talk about a lot was we're trying to cure a disease where one of the biggest known kind of effects of the disease is degradation of memory. But we don't know what memories are. We don't know where memories are stored in the brain. That's still a very highly debated field. We don't know how memories are retrieved. So we don't fully understand memory. So we're trying to cure a disease where the biggest impact from someone who has the disease, one of the things that people are kind of most afraid of when we think of dementias and Alzheimer's is the loss of memory. But we don't know what a memory is. So how do you cure something that you truly don't understand what that piece of the puzzle looks like? Okay. And so all of these things are more difficult, and we keep shifting and better understanding. We see a disease progress, but what happens before we knew that disease was there? And so that's something we're starting to see more now in medicine and science is we can categorize what a disease looks like once we know the disease is there, but was there something happening 10 years before that that's consistent? So we see that in Parkinson's disease, about 80% of Parkinson's patients present with GI issues 10 years or more before they're diagnosed with Parkinson's. So is that actually part of the problem? Is that part of the story? We're seeing that more and more and more disorders, that we're trying to look back before you had this disorder. Was there something happening in your health that could have told us this was coming? Are there early symptoms we can treat before we get to the part where, the disease is really debilitating? And so now we're looking at kind of as the body as a whole, we're able to see that there actually may be trends in people that present with these kind of these diseases, that maybe there were symptoms beforehand. Maybe the disease is actually starting much earlier, and we could be treating there, and maybe that impacts these later things. And so science is a really exciting place all the time, but it's really exciting right now because there's this growth in kind of understanding whole system. And I think by looking at the whole system, by looking at large amounts of data and putting them together from different fields, I think we're able to ask better questions. And I think we're able to kind of fast forward a little bit, because looking at what's happening in a bunch of different ways sometimes actually makes the picture a little clearer.
Exactly. So I'm assuming, like right now, because we said that there are many factors that happen to our different cells, but let's say like for a baby, if we teach a baby's cell types to like an AGI or like a large language model, it should be able to guess basically for the entire humanity what are the cell types, maybe not the effects that, like, for example, sports have on the cell, like on different cells because they mutate differently. But I'm just saying, like, as the default, like if we want to know the nature of the cell, I think that's going to be possible very soon, right?
Yes and no. In early development, I'm going to say when I studied this, but I did a lot of brain development, which is part of how I got into Alzheimer's. But basically, you sometimes, in some cases, in some areas, you have more when you're younger, and it prunes down to be best functioning for the environment you're in or the system that's happening. So we talk a lot about, like, certain things have to happen at certain times so the body progresses through development. And if that's missed, basically the system rewires to associate that didn't exist. So you see this a lot. The best example from my mouth would be language. So if you're never exposed to language, your brain rewires around the concept of language because it wasn't used. So cell types change, wiring changes. So it's not as linear as, like, so as a baby, they have cells, nothing else has happened. Well, I mean, in utero, a ton of things happen. What the mom eats, what environments, what kind of, toxins and stuff we're exposed to. Before that, the age of the mother impacts it, because that will change, that could have impacts on how the cells divide and stuff to make a new life. So that can impact it. So, like, where is the purest form? Where is the, like, nothing has happened yet, so we can use this as our template, is even harder, because where that is, I don't think we truly know. And we know very little about what's happening fully in utero in terms of humans, because there's a lot of rules and regulations around that. So there's still a black box really in what's even happening and how you go from, the beginnings of cells dividing kind of to the end to having a human life. There's some unknowns there. So we're still kind of learning about that. So, like, there's very few truths that we know everything about in science, because as we learn more, as new technologies come up, it really just shows us how little we know. And then we kind of grow with, our access to knowledge and our ability to see things. And so we talk about AI, like AI will change science. And because of that, we will get more questions, and that will allow us to then grow our knowledge base. But every time something like this comes along, I mean, like the invention of the microscope changed everything. We're now able to ask better questions and kind of fill in kind of some of the blank spaces. In some cases, we know those exist. Sometimes technology shows us that actually we missed a whole section of the puzzle. And so we have to go back and fill those in. So it's never as linear as you want it to be.
This is very interesting. Thank you so much for your time. I hope we can do like a part 2 of this, because like we're running out of time now.
Yeah.
But this was very great. Thank you so much.
No problem.
Take care.
All right.
Bye, everyone. Thanks for watching.